I've been asked to talk about differences in CAR T products, and what I hope to do today is kind of take it back a notch or two, just so that everybody is on an equal footing, no matter what your role is in the patient journey for delivery of CAR T. So I'm trying to move my presentation on a moment. I hope that's moved. Toby, please let me know if it hasn't. So what I just want to cover today in brief is just to cover what T-cell activation actually is, what the T-cell receptor and co-stimulatory signaling actually does for a T-cell, and then have a bit of a better appreciation for what actually makes up a chimeric antigen receptor. In that way, then we can kind of cover how some of the different products vary from each other. And then that'll give us a better understanding about how we think the CAR T expansion kinetics vary between the products, and then potentially speak to how we think some of these might be resulting in different toxicities after the delivery of CAR T products. And then just at the end, I'll give a bit of a nod to the evolution of CAR T in the future. So very much sort of based not specifically on one product, as I know a lot of this will be covered in other talks, and trying not to veer too much into how we manage the toxicities either. So if we start, then apologies for lots of people who will already know this in great depth, but it's important, I think, to acknowledge that native T cells can only really recognize snippets of proteins that have been processed by antigen-presenting cells or some stroma, and then those snippets of protein are presented back to the T cell in the context of HLA and proteins. And the T cell receptor recognizes both that pitched up bit of protein, the antigen, and also the HLA protein. And as a result, when naive T cells are circulating around the lymphatics or the blood, then these such signals might keep the T cell alive, but it won't activate them to actually do anything about it. And in order to be activated and to divide, a naive T cell definitely requires another second signal or a co-stimulatory signal. And classically, CD28 is another protein on T cells that does this very effectively. And when a cell receives both a TCR signal and a CD28 signal, then they're licensed to go and be activated and divide. In the case of memory T cells, I think it's important to reflect that these are cells that have already seen their antigen or their snippet of antigen once before in their life. And as a result, they are more effective at responding a second time. They persist in the absence of antigen afterwards, and this is the whole basis for performing vaccination. That's why it's effective. And there are different sets of memory T cells that generally reside in different tissues of the body. And they utilize different sorts of second signals. So whilst both memory and naive T cells will rely upon that T cell signal from the T cell receptor, they will utilize different signals to expand as their co-stimulatory or second signal. So I've already mentioned CD28 as being a very important signal for naive T cells, where it will promote the cell survival and promote the differentiation and activation. But a other type of molecule called 4-1BB can do the same, particularly for memory T cells. 4-1BB is actually a herpes virus receptor, and it's related to more chronic stimulation in the periphery. And it can allow a T cell to survive, allow it to divide, allow it to produce cytokines in a similar way to CD28, but perhaps not so much as an explosive initial activation profile. And I think that's important. So just to pull all this together, a normal T cell will require the T cell receptor signaling. It will require a second signal of some sort, and then its response can be propagated by cytokines either directly from the antigen presenting cell or from elsewhere. In contrast, the B cell receptor is effectively a membrane-bound antibody. So we have antibodies circulating around in our blood, but the B cell receptor is tagged onto the surface of the B cell. And the key difference here, as I'm sure you're all aware, is the fact that the antibody variable regions here are able to recognize the native protein itself. The antigen does not have to be chopped up and presented on HLA antibodies and proteins. So in that way, it's a very powerful receptor. And the concept for CAR T therapy was to combine the power of this receptor with the transduction signaling of the power killing, the tumor killing power of the T cell. So that's effectively what we try to do. We're genetically modifying patients' T cells so that they can recognize the native specific proteins on cancer cells without the need for HLA presentation, and then allowing CARs to be effective killers. And as a result, they are considered, I think the concept is really important, that they are a living drug because they persist for long periods of time afterwards. And as has already been referred to, I'm sure the patient needs to have these T cells isolated from them, and then they go off to the relevant company involved, the relevant pharma company, to be engineered. And this is a step that can vary between companies and even between products, as has been mentioned. And I will just explain a couple of those in a minute and just tell you what the different processes are. Those modified T cells are then grown and expanded in culture with the different companies. And then those products are then brought back as bona fide pharmaceutical drugs and then reinfused back into the patient. And David will go into that process in more detail in the next talk. So in terms of the design of the chimeric antigen receptor, like I said, we're using the antigen specificity of an antibody and combining it with the transduction and the activation power of the T cell receptors. And this is possibly a bit nicer to see and look at. So here is a design for a third generation CAR. The first generations had the antigen binding domain, a hinge region to get over the cell membrane, and then combined it with just the CD3 part of the T cell receptor to allow the signaling. And then the second generations and third generations have incorporated co-stimulatory domains to directly give both the T cell receptor and the co-stimulatory signal once the native antigen has been bound by the antibody. And in that way, recognition of the tumor protein can directly activate the T cell, irrespective of any presentation by any other cell. Now, there is variety in the products, therefore, that are produced. And this slide just shows 3 of them, which are in common use. So these are all the CD19 targeting CAR T products, which are used in treating lymphomas, for example. So this first one here was developed at NCI and is the Kite Gilead product, so Axi-cel or Yescarta. And here we have the UPenn product, which was taken by Novartis and produced now and named as Kymriah. And then we have the Juno Therapeutics Lysocell product as well. So with all of these, you can see that the actual variable antigen responsive domain, the antigen recognition domain here, is common between them, and they're all using domains that recognize FMC63. In terms of the hinge transmembrane, the thing that gets the signaling across the cell membrane, both the Kite Pharma and Juno Therapeutics adopted the CD28 transmembrane domain, but Novartis opted for a CD8 alpha domain, a different protein. Then importantly here, the first signal is always the CD3 zeta chain, so transmitting that TCR signal into the CAR T cell. And the second signal has varied between these products. So Kite Gilead and Axi-cel employs the CD28 co-stimulatory protein, as has been mentioned in previous talks. But other products have used, so Novartis have utilized the 4-1BB co-stimulatory signal, as have Juno Therapeutics. And the way in which these have all been transduced into the T cell varies. So Kite use a retroviral program, whereas Novartis and Juno have employed a lentiviral program to get the genetic transduction of the T cells optimized. And this may change again in the future as different processes come online, such as CRISPR technology. This is a really busy slide, and it just goes to show you that having had a few products that are in use at the moment and in trial, when it comes to the myeloma products, which will be a step again away and is coming through pipelines and trials, We can see that there are a plethora of products, all using different transduction mechanisms, all with different sorts of variable fragments recognizing BCMA as its main target, and with a variety of different co-stimulatory domains again being utilized. So this is a major way in which CAR T products will vary. And we think that these differences can result in different kinetic profiles after infusion of the CAR T products. This is just a mathematical model, so perhaps the pharmacists in the audience might be a bit happier with this sort of graph than many others, but it just proves the point that after infusion of CAR T, we expect to have a little decline in the CAR T numbers initially in the first day or two, and then there is a huge expansion as the CAR Ts come into contact with their tumor in vivo. And this expansion is maximal very quickly. And we reach this Cmax, as you see here at the top, at the latest, I would say, at about 14 days. And then over time, this actual peak number of CAR T cells wanes. This data that I'm showing now actually refers to the peak CAR T cell monitoring for axi-cell. So I think, as you can see here, as utilizing a CD28 construct, we see a more prompt peak expansion of CAR T cells, which is manifest around the 7-day mark. And we think that this then encourages the manifestation of toxicities. Co-stimulation, therefore, probably plays a major role in modulating this CAR T expansion and their persistence long term. And this is just a kind of demonstration of what you might expect to see in terms of those pharmacodynamics of the CAR T expansion with a CD28 second co-stimulatory construct and with a slower acting 4-1BB construct. And certainly, we are now getting emerging data to show that peak CAR T levels are associated with toxicities. So this data is courtesy of Kite Gilead and recently presented. And you can see that for patients, the few, admittedly, patients that get grade 3 or more cytokine release syndrome have a higher peak CAR T level compared to those who do not. And certainly also for those patients who have grade 3 or higher neurotoxicity, there again is a higher peak CAR T level. This obviously is data pertaining to axi-cell, but I think, as Toby was saying earlier, may also apply to Tecartus. This is a table it's a horrible table, my apologies, and I am to blame, I put it together. But I hope you'll be able to see in time that there are key differences then between these products and the overall time and grade of CRS and ICANS. I think it's important also to note that Kymriah, for example, can elicit different CRS rates if administered for lymphoma versus ALL. And the other 4-1BB co-stimulatory domained products, such as Breyanzi, also have a similar rate of CRS. In contrast, as has been mentioned, Yescarta, so that's axi-cell and Tecartus, have the highest rates of CRS, being detectable at all grades in north of 90% of patients. And as has already been mentioned, this happens very early on with that expansion. So as these T cells undergo that explosive expansion in the patient, they release their cytokines and the patient looks as if they are septic with low blood pressure and low oxygen and high temperature. Similarly, then, the timing to ICANS is a bit shorter in contrast to the 4-1BB constructed products, and the duration of ICANS can be slightly longer, and a higher proportion of patients have any grade of ICANS. I think what's also becoming clear now with regards to differences between T cell products is that the total number of T cell subsets within a product can differentially associate with the toxicities that are manifest. And of course, this then takes that risk back to a per patient basis rather than a per product basis. So again, this data is pertinent to axi-cell. And we can see that patients who had grade 3 or more ICANS had a greater number of effector T cells present in the product, and those that had grade 2 or more cytokine release syndrome had a greater number of central memory T cells in the product. I've written a note here to remind myself quite aptly that actually there are other differences in the manifestation and the clinical spectrum of ICANS in particular that can be as a result of the target of the CAR T. So CD19 is a wonderful target for CAR T and for other immunotherapies because it is exclusively presented by B cells and B cell tumours. However, BCMA, which is a prime target in myeloma and being used in many CAR products, is actually also present on peripheral nervous tissue and importantly in the basal ganglia of the brain. And as a result, within the trials that have been conducted so far, it's clear that the clinical spectrum of ICANS for BCMA CAR T includes potentially Parkinsonian syndrome, which at its worst can be lethal. And that, I'm sure, with time, will result in different protocols for the treatment of ICANS with that product. We also are seeing more data now to suggest that the composition of CAR T products not only affects the toxicity, but perhaps also the efficacy. And CAR T products that have T cells that appear more tired or exhausted seem to have a poorer response, whereas those products that contain an increased proportion of naive T cells seem to have improved event-free survival. And again, this data is pertinent to axi-cel. So overall, I hope I've shown you that there are pertinent differences in the structure of these CAR Ts, how they're made, that have real differences and pertinence for the patient. And these are all in evolution. So we have started out with first generation CARs just with a T cell signal, then incorporation of co-stimulation, and then next generation CARs, which are going to incorporate more signals and generate more memory responses and hope to allow persistence of the CAR. Other ways in which CARs might be modified in the future might include different variable chain designs or using different domains to put together a different product, using different vectors or methods of getting the construct into T cells, as I mentioned before, having multiple targets to the CAR, so using CD19 and 20 to try and overcome some of the mechanisms of disease relapse, which can include loss of the targeted CD19 antigen with regards to B-cell lymphomas, and varying the modifying cellular composition of the product, as we've seen here, some data that might be very effective, and armoring CARs so that when they are activated, they produce other modulators and other ways of killing cancer cells directly, increasing cytokine production, for example. And this is just to scare us all, I think, this last slide of mine, which is just to say that there is an enormous variety of CAR T targets being studied. And as we get going with our heme malignancies, in the back of our minds, obviously, is an awareness of the plethora of different targets which could come online in the near future. And of course, all of these products will be unique with their own targets, potentially with their own clinical spectrum of toxicities, and with their own constructs and variabilities. So I'm going to leave it there for the sake of time. And I hope that that has given some insights. And I'm happy to take any questions.